Today we'll start a new chapter. It's about the active site. Okay. Now we will take in more in details what is the active site and how we can identify the amino acids involved in the binding and in the catalytic site. So previously in the first, second or third chapter, we discussed that the active site is a cleft at the surface of the protein of the enzyme, and it contains mainly the binding site and the catalytic site. It's where the reaction, the substrate will bind and where the reaction takes place. So, and when we discussed about the active site as binding or catalytic site, we said the site is a side chain of a specific amino acid involved in binding or in catalysis. So now in this chapter, we will discuss how we can identify these amino acids involved. This is the main focus of this chapter, how you can identify these amino acids who are involved either in the binding or in the catalysis. Okay, so what are the methods we can use to identify these amino acids present in the active site, or if we can, for example, say this amino acid is part of the active site or it's a amino acid outside the active site, etc. So first, in this chapter, we'll discuss the investigation of the 3D structure of the active site and how we can identify the binding and the catalytic sites by different methods. We have the trapping of ES complex. As you know, as we said previously, you have the first step of the reaction E plus S to ES. It's a reversible reaction. But the second phase from ES to E plus P, it's very a quick reaction. So once you have the formation of ES, the breakdown of ES to E plus P is very quick. So it's very difficult in a enzyme catalyzed reaction to isolate the complex ES, because once you form ES, directly it will dissociate to E plus P. So we will find, we will see and we'll discuss in this chapter some methods to trap ES in its a specific form as ES. And here we will see we may not use the substrate itself, but something else. Or some chemical process that we can use to trap ES complex in its form as ES to be able to isolate this interaction between the enzyme and substrate to know what are the binding sites, what are the amino acids involved in the binding. And we have some methods by using substrate analogs. We can trap the enzyme substrate complex. We can use some irreversible inhibitors as well to identify the amino acids. We can use enzyme modification by chemical procedures affecting specific side chain of amino acids. We have treatment with proteases. We have the side-directed mutagenesis when you do a mutation for a specific amino acid and you see the effect of modifying a specific amino acid by another. What this will affect the enzyme as kinetic parameters Km and K catalytic, and we will see how we can evaluate the binding site by variation of the pH. So this in general is the chapter. It's a very important chapter to identify the amino acids involved in the active site. And it's in general, except the last part, which is the effect of the changing pH. The first part of this chapter, it's more something you need to analyze rather than drawing plots. So in the first part of this chapter, you don't have to draw any plot to analyze or to do your exercise or to analyze the given last part about the pH. Here we continue with the kinetics and you have to draw some plots. So in this chapter, we will take a break from drawing plots, mainly in the first part. But sometimes this is more difficult. And plots is easy to do. It's easy to identify parameters from the plot. Here you have to analyze more, you have to read, you have to understand, and you have to analyze the given to obtain data about the binding and the catalytic site. So it's from one side, you don't have to do plots, so you have to analyze more in this chapter. So keep in mind that in the enzyme catalyzed reaction, you have a important step, which is the formation of the enzyme substrate complex, which is the first step in any enzyme catalyzed reaction. So the catalytic power of enzymes comes from their bringing substrate together in a favorable orientation to promote the formation of the transition state in AES complex. So ES, a transition state. So this is very important in any enzyme catalyzed reaction. So the substrate will bind in a specific area in the active site called the binding site. At this active site, and most enzymes are selective. They are highly selective in the substrate they bind, and this you know from the second chapter. And the catalytic specificity of enzymes depend on the specificity of binding. So what is the active site? In general, the active site is a region that binds the substrate. It contains the catalytic group and promotes the formation of the transition state. A cleft present at the surface of the protein where the substrate will bind, where the catalytic reaction will occur, and for sure, it's the area that promotes the formation of a transition state. And keep in mind that the active site contains amino acid, different amino acids, very close to each other in the 3D structure of the enzyme. But if you look to the primary structure, as it's seen here in B, if you look to the primary structure of the protein, you can see that these amino acids close to each other in 3D structure of the protein of the enzyme, they are far away, they are distributed far away in the primary structure. Okay, so منهم بالبرايمري structure 35 36 37 38 حد بعض. بس هالفولدنج للبروتين خليهم كل هالامينو acids اللي هن involved in the active site to be close to each other in a 3D structure. So keep in mind that the active site have common features. One of them, the active site, is a three-dimensional cleft formed by groups that come from different parts of the amino acid primary sequence. So the active site may include distant amino acid residues present in a primary structure, as I just said. You can see here in the figure A, the upper part of the figure, you have the 3D structure of, or the Rabin diagram of the enzyme, a lysozyme, with several components of the active site shown in different color, in yellow, in green, in red, in orange, etc. These are the amino acids. Here is the active site of the enzyme. However, if we look to these amino acids in a primary structure, you can see in the primary structure, you have these residues are in different part of the polypeptide chain. Second, the active site takes a relatively small part of the total volume of the enzyme. So it's a small cleft present at the surface of the enzyme. Keep in mind that enzymes, they are protein, they may have for sure more than 100 amino acids, and they have a diameter more than 25 angstrom. And not all these amino acids are involved in the binding and in the catalysis. Only the amino acids present in the active site are involved in this. But keep in mind also that we have some amino acids not involved in the active site, but they are very important in the folding and formation of the 3D structure. So even if you lose these amino acids, you may lose completely the activity of the enzyme because these amino acids are very important in the formation of the 3D structure of the active site itself. Okay, so some amino acids serve as a scaffold or regulatory sites to create the 3D dimensional active site from amino acids that are, as I said before, far apart in the primary structure of the protein. So active sites are clefts substrate molecule are bound to a cleft. Once you have this binding, the water present in this area will be excluded unless water is a substrate, is a reactant. And then you have non-polar character of much of the cleft that will enhance the binding of the substrate as well as the mechanism of catalysis. So once the substrate will bind, the water present in the cleft will be excluded. And in this case, the hydrophobicity of this area will decrease, and this will enhance the binding, will help the binding and the catalysis. And also this is something we discussed before, that substrates are bound to enzymes but by weak interaction. There is no covalent mode present between the enzyme and the substrate. Non-covalent interaction are present to form this ES complex. So they are weak non-covalent interactions such as the electrostatic interactions, the hydrogen bonds, the van der Waals forces, the hydrophobic interaction, ionic interaction. All these interactions, they are present between the enzyme and the substrate. So you can see here the dots present in your in the figure are hydrogen bonds between the enzyme and the substrate. So what are mainly the function of the catalytic residues? The catalytic residues are directly involved in the mechanism. For sure, they alter the pKa value of residues directly involved in the catalytic mechanism. And this later on when we will discuss the pH. These residues for sure stabilize the transition state, the intermediate ES and F in transition state. And for sure, they activate the substrate to be able to be transformed to product. So in order to understand how the enzyme work, it's very important to know not only the amino acid residues involved in the binding site and in the catalytic site, but also it's very important to know the environment in which each site exists. شوفي حواليه, okay? And how the arrangement and space of the sites with respect to each other. لأنه نحن بنعرف إنه بدنا يكونوا aligned the binding and the catalytic site with the orientation site. They should be aligned in order the reaction to take place. So what are these amino acids present in this enzyme in general? Okay. We can summarize that an enzyme contains, in order of increasing functional importance, different type of amino acids. One, we can call some amino acids as non-collaborative. What do we mean non-collaborative amino acids? They are present for sure in the protein. Their role is unknown. They can be removed without reducing the activity of the enzyme. يعني this enzyme contains a lot of amino acids. إذا جيتوا عملتوا hydrolysis of part of it أو إذا عملنا mutation for several amino acids, هول الامينو acids ما بيغيروا بالactivity for the enzyme ولا بالbinding substrate to the enzyme. So these amino acids are called non-collaborative. Another type of amino acids called collaborative. What do we mean by collaborative amino acids? These amino acids, they support the functional amino acid. We can remove them. The enzyme activity will persist, but we can notice that the enzyme becomes a little bit weaker. So مش إنه هن ما عندهم ولا role وبنقدر نشيلهم without any modification in the activity of the enzyme. صح ممكن ينعمل لهم mutation أو تغيرون وتضل الactivity constant, but you can notice that the enzyme becomes weaker because their role is to support the functional amino acid in the enzyme. We have a third type of amino acids present in this enzyme that we call them the auxiliary amino acids. These amino acids do not interact directly with the substrate of the reaction. However, they provide mobility for areas surrounding the active center, and they provide the molecular chain flexibility. And these amino acids are very important in the formation of the active site itself. So لانه ما ننسى نحن الانزيم once the substrate will bind في عندنا modification in the structure of the enzyme مش in the structure of the substrate. So في عندنا هالflexibility المولكيولر chain of the protein. So مين عم بيقدر يعطينا هالflexibility؟ هن هالauxiliary amino acid important for the formation إذا بدكم of the 3D structure of the active site. It's important in this flexibility. بس هول كمان هن منهم component of the binding site and the catalytic site لأنه fourth type of amino acids اللي هن in contact, their role is very important. They are component of the active site. They are very close, a very close distance with the substrate, and they are in direct interaction with the substrate itself. هول اللي هن components of the active site اللي هن binding and catalytic. هول بنسميهم they are in contact amino acid with in contact and in contact with the substrate. So we have these four type of amino acids present in the protein. أكيد الauxiliary in contact are very important for the activity of the enzyme. يعني ممكن نشيل هلا حنشوف كيف difference and how I can differentiate if an amino acid is in the active site يعني هو part of the in contact amino acid ولا هو auxiliary amino acid important in the 3D structure of the protein. Later on, with our examples and with our methods, we can differentiate about these amino acids. So here, as you can see, في عنا a cartoon representing a substrate and an enzyme. في عنا كل شيء شايفينه بالأزرق هن side chain of amino acid not interact with the substrate, okay? اللي شايفينهم اللي هن in orange هن اللي directly in contact with the substrate واللي شايفينهم الأخضر هن اللي بيعطونا هال 3D structure of the enzyme and the flexibility of the active site. يعني في عنا هون اللي شايفينهم ال in contact، عنا شفنا ال auxiliary and the non-collaborative أو amino acids اللي ممكن نكون عم نشيلهم بلا ما تأثر على ال activity of the enzyme. Keep in mind that an enzyme free as a structure is different than an enzyme bound to the substrate. هيدا على طول بدنا نضلنا مذكرين انه once the enzyme the substrate binds to the active site of the enzyme, you have some modification in the structure of the enzyme to be able to take the shape of the substrate to form this complementarity between the enzyme and the substrate. So if you take a structure of the free enzyme and the structure of the enzyme bound to the substrate, you have different structure for the enzyme. Okay, here you have the carboxypeptidase A in its free form and its form bound to the substrate. As you can see, this slight difference in the structure when the enzyme is free and when the enzyme is bound, and this gives an importance for these auxiliary amino acids, important for the flexibility of this protein. So a very important role in the active site here, three side chain of the amino acids important or they are involved in the active site. They are very important amino acids اللي حكينا عنهم قبل كتير. في عنا amino acids involved in the binding, في عنا amino acids involved in the orientation, and we have the amino acids involved in the catalysis. So you have this catalytic triad. We should never forget about a catalytic triad important in the catalytic site or in the active site of an enzyme for the reaction to take place. We have here an example for the chymotrypsin where you can see the enzyme, the chymotrypsin with a substrate in the active site of this enzyme. You can see that you have the histidine here present involved in the binding for the substrate. You have the hydroxy group of a serine that's involved in the orientation, and you have the tryptophan also present in this active site that will ensure the activation and the catalysis of the reaction. So I have this catalytic triad for chymotrypsin which is serine, the histidine, and tryptophan, important in the as catalytic triade, important in the mechanism of chymotrypsin. Chymotrypsin, keep in mind, since it contains the serine, and we will see later on a lot of enzymes where the serine is involved in the catalytics in the active site. These enzymes are called serine proteases, and we will see a lot of protease منهم كمان غير الكايموتريبسين. You will see also the trypsin. في عندها السرين involved in the active site, and all these proteases will be called later on serine protease because they all include the serine in the active site of the enzyme, and it's very important in the catalytic triade. That's why they give them the name of serine protease. One amino acids which is very important in the active site. يعني إذا نحن عم نروح نفتش هلا على amino acids involved in the active site of a protein, one of the amino acids that you always have to look into it if you have any doubt about its presence in the active site is the imidazole ring of histidine. Well, this is very important because the imidazole side chain of histidine might be expected to be an important contributor towards the catalytic activity, since it's the only amino acid side chain to have a pK in the pH range at which most enzymes function. In fact, the histidine or the imidazole ring of the histidine act as both a proton donor and acceptor, and this make of these amino acids as important amino acids in the active site of any enzyme. So if you have any doubt that a histidine could be present in the active site, directly you have to test this hypothesis. لأنه yes, there is a big possibility that the histidine could be involved in the catalytic triade because this important characteristic as donor and acceptor of electron or proton. The imidazole ring transports acyl groups and phosphoryl groups. It participates in the activity of a lot of esterases, nucleases, and some proteases also. And it's found in oxidative phosphorylation, in the transmission of the nerve impulse of the neuromuscular junction, and in a lot of in the role of muscle contraction. So after this first introduction about the active site that you know most of it in general, the question that we have to ask now, how we can determine these amino acids involved in the active site. يعني أنا ما بكفي أقول عندي هيستidine عندي سيرين، بدي أقول أنا سيرين قد إيه رقمها بالبرايمري سيكونس. يعني أنا عم بقول سيرين نمبر 96 أو أنا عم بقول histidine number 210. يعني I have to specify the amino acids in this protein sequence that they are part of this catalytic triade or they are part of the active site. So for sure, the big question, the main focus of this chapter will be how we can determine these amino acids involved in the active site. What are these methods that we can use to identify these amino acids? Before we continue, do you have any question till now, please? No questions? Okay. So one of the methods, which is very important method to use in the identification of the amino acids involved in the active site, is trapping the enzyme substrate complex. يعني trapping يعني أنا يضل ال ES as a complex لا أقدر أعمل له isolation. In fact, once you can isolate the ES, you can study what are the amino acids involved in the binding between the E and the substrate. إذا ما قدرت أعمل له isolation ما بقدر أعرف شيء، ما حأعرف كيف كانت الانتراكشن between the enzyme and the substrate. So we can do this by some chemical process, and we will see what we mean by chemical process. So as you know that the reversible character of the steps involved in the enzyme catalyzed reaction makes the determination of each substrate binding site less than a straightforward step or mechanism, because it's very difficult to isolate the ES as it is. At steady state, a constant amount of ES complex is known to be present, yes, but if you try to isolate this complex, and then you have to once you isolate it, you can hydrolyze this bonds, present and whole weak bonds, to identify the amino acids in which the substrate is attached to. If you will try to do this, all your effort will not be rewarded with success, because the substrate will dissociate from the enzyme during the procedures involved in the isolation. So you will not be able to isolate naturally the ES complex. Okay. However, we can trap the ES. The ES complex can be trapped, but in this case, it will be trapped in a modified form by using some chemical process. So the substrate is no longer able to dissociate from the enzyme. And in this case, you will be able to identify the substrate binding site. We'll take an example for the modification by chemical process or the trapping by chemical process, and we will take the reaction catalyzed by fructose biphosphate aldolase. This reaction is interaction between the dihydroxyacetone phosphate and glyceraldehyde 3-phosphate to give fructose 1,6-biphosphate. This is one of the reactions that you will see in the neoglycogenesis or in glycolysis. You will see it later in metabolic. And the main importance now to see how we can trap the ES complex. Okay. Researchers showed that if the reaction mixture, yani substrate, is treated by sodium borohydride, full characteristic لها sodium borohydride, it's a strong reducing agent. This will help to form an inactive complex that we can isolate. شو يعني؟ نشوف نحن الرياكشن. عندنا الانزيم، هلا انسوا انه في عندنا NH2 بالسايد تشين involved بالانزيم. في عندنا الانزيم وفي عندنا السبستريتس اللي هن جليسرالديهايد 3 فوسفات ودايهيدروكسي اسيتون فوسفات. بالميكانيزم اذا بنطلع انه حيصير في عندنا شوف بيز as a complex، وهيدا الرياكشن هي اللي رح تصير، لانه هلا بعدين عرفنا انه في عندنا NH2 حتصير الانتراكشن بين ال NH2 والCO. نورمالي هالرياكشن رح تكفي اوكي as ES complex مع الجليسرالديهايد to form the fructose 1,6-biphosphate. هيدا النورمال procedure للرياكشن. When you treat this reactional mixture with a reducing agent, then ABH4, this bond will be reduced. And in this case, directly you will have this NHCH by adding this reducing agent, and this will form, instead of a ES complex active to give product, an inactive complex that will not be able to give product. And in this case, you trap, هذا اللي اسمه trapping, you trap the ES complex in another inactive form. And now, once you have this inactive form, you can isolate it, you can hydrolyze the interaction between the enzyme and the substrate, and you can study what amino acid was an interaction with the substrate. You can do a sequencing to know the amino acid involved in the interaction with the substrate. And using this method, a lot of enzymes or amino acids involved in the active site of enzymes were identified. So by chemical process, we can identify some amino acids involved in the binding site with the substrate by using uh, reducing agents, more specifically the NABH4. So once the AS complex has been trapped as its inactive complex, it will be subjected to partial hydrolysis, and the amino acid sequence for a few residues in each side of the binding site will be determined. And on hydrolysis of this inactive complex, they were able to identify the N-glycerol lysine found among the products. And from this, they were able to conclude that the dihydroxyacetone phosphate normally binds to a side chain amino group of a lysine residue in the enzyme, and this form a Schiff base, the linkage, يعني N double bond CH. And this was reduced by the reducing agent and an ABH4, and this induced the formation of an inactive complex to, as I said before, help to isolate, to trap the enzyme in this form and to identify the amino acid, which is the lysine in this case. This is first method. So you can use biochemical process to trap a enzyme substrate complex. We have other methods, is the use of substrate analog. One alternative way of producing a complex more stable than the normal ES complex is to replace the natural substrate of the enzyme by an analog which binds to the same site of the enzyme, but in this case it's less rapidly removed. ما ننسى يعني مثلا part of these analogs they are known as competitive inhibitor for the enzyme. Okay, they will bind to the enzyme بس بدهم وقت أطول يعملوا dissociation لأنه not the normal substrate for the reaction. هيرجع يعطوا E plus S بس بدها وقت أطول من ما كان ES يعطي E plus B. We'll see one of the analog used for chymotrypsin, هو paranitrophenyl acetate. So the first step في عنا نحن الانزيم مع paranitrophenyl acetate هيعطينا acyl enzyme plus paranitrophenol. This acyl enzyme كمان هون as a complex E مع الانالوج للسبسترate is not the natural ES complex, and this will form a inactive complex that also could be isolated and analyzed. So the first step in the hydrolysis of the paranitrophenyl acetate and other acyl esters in general by chymotrypsin is the rapid splitting of the ester to yield the first product, the alcohol, and to form an acyl enzyme. The second step is the liberation of the acyl group from the enzyme, which is in this case extremely slow, allowing the structure of the complex to be isolated and to be investigated. And they found that the acyl group binds by an ester linkage to the OH group of a serine present in this enzyme, and this serine is the serine number 195. And they were able to isolate or to identify the serine 195 as an important component for chymotrypsin in the active site, one of the catalytic triad of the chymotrypsin. So using analog for the substrate, they were able to trap a ES complex in another form and analyze this ES complex to identify amino acids involved in the triad. Other methods that we can use also is the use of irreversible inhibitors. As you can know, already by irreversible inhibitors, these are inhibitors that will bind to the enzyme, but they will not dissociate, and they will trap EI as EI. Okay, so one of the methods to trap or to analyze this is to use these irreversible. These inhibitors, they will bind by covalent bonds to the enzyme, and they provide an alternative and sometimes complementary means for studying the functional group at the enzyme active site. because they modify the functional groups which can be identified in this. For irreversible inhibitors, we have different categories of irreversible inhibitors. We have these categories are mainly three. The first one is called a group-specific reagents. The second is called affinity labels. And the third is called the suicide inhibitors. These three categories belong to irreversible inhibitors. But what is the difference between each category? The first one, the group-specific reagents, whole irreversible inhibitor, okay? So with difference بيناتهم in how they react with the enzyme. These group-specific reagents, they react with a specific side chain of an amino acid. يعني نسميهم جروب سبيسيفيك، يعني انا هيدا الموليكول او هالانهبيتور interact with a chiol group SH group. يعني انا بعرف انه اذا عندي SH group chiol group يعني عندنا cysteine involved اذا عامل معه interaction. Okay, so wholly group specific, they are going to react with a specific side chain of an amino acid. Okay, هو اللي بنسميهم group specific reagents. We have the second category, affinity labels. These are molecules that are similar in their structure to the substrate for the enzyme, but they covalently modify the active site residues. They are more specific to the enzyme active site than the group specific reagents. يعني انا group specific reagents اذا حطيته as inhibitor in my medium بده يروح يشتغل، بده يروح يعمل interaction with the side chain of any amino acids اللي هو بيعمل reaction معه. However, the affinity labels in the molecule عندهم similarity with the substrate itself، بيعملوا covalent bond محل السبستريت بالاكتيف سايت، هو اكثر specific للانزيم itself لانه عم نحكي عن substrate like molecules. However, the third category which is the suicide inhibitor هو كلهم هلا حناخذهم one by one, okay, with some examples. The suicide inhibitors are mechanism-based inhibitor. Okay, they are modified substrate that will provide the most specific means to modify an enzyme active site. These inhibitors will bind to the enzyme as a substrate. The first mechanism of the reaction will work normally, so the initial procedure of the mechanism will go as normal as a substrate. However, the mechanism of catalysis will produce an intermediate who will inactive, okay, intermediate during the catalytic mechanism. يعني أنا بكون عندنا مش مثل الافينيتي ليبل أي أنالوج اللي عنده similarity in structure بيعمل binding وخلص بوقف هون، لا هو مثل السبستريت بيعمل binding، الرياكشن بتبلش بس once the reaction starts you form an intermediate. هيدا الانترميديت during the reaction بيكون inactive. Okay, it's a reactive intermediate that will inactivate the enzyme through this covalent modification. So these are the three different type of كلهم irreversible inhibitors that we can use to identify amino acids of active site. Do you have any question? Okay, so just a small reminder that all these group-specific reagent, affinity labels, and suicide inhibitors belong to the category of irreversible inhibitors. لأنه بعدين تنسوا إنه هو irreversible inhibitor. ضل براسكم إنه هو ثلاثة categories are irreversible inhibitors. We'll start with the first type, which is the group-specific reagents. So these are irreversible inhibitors, as I said before. They are very useful in the investigation of the active site. Since the inhibitor here, unlike the substrate, منه substrate will bound to one specific side chain of an amino acid present in the active site of the enzyme, and it will act as a marker to enable it to be identified. So one of known group, for example, in the organophosphorus compound will react with essential OH يعني of a serine side chain of some enzyme ممكن هنشوفهم بعد شوي. عندنا DIPF which is a nerve poison, since one of the enzyme it will inactivate is the acetylcholinesterase, important in the nerve function, and that we will see in a little bit. So we will see some group-specific reagents. For example, you have اللي حكينا عنهم هو هلا الاسيتيلكولينستريز اللي عنده as amino acid in the active site the serine اللي عندها OH group، وفي عندنا نحن جروب سبيسيفيك رياجنت اللي هو الدي اي بي اف داي ايزو بروبيل فوسفوفلورايدات اللي هو a group specific reagent that will interact على طول with a OH group hydroxy group of a serine. So هيدا المولكيول once you add it to a enzyme وعندها ال OH group عندها serine residue هيدا الدي اي بي اف او دي اي اف بي بتلاقوها باثنين abbreviation will react directly with the hydroxy group of a serine to form a inactive enzyme. So in this case, the DIPF can inhibit an enzyme by covalently modifying an important crucial serine residue present in the active site. وهيك صار عندنا الانزيم inactive. So إذا صار الانزيم يعني إذا أنا عندي الانزيم لحاله مع substrate حطيته وعطاني رياكشن النورمال رياكشن، جيت انا حطيت الانزيم مع دي اي بي اف وحطيت السبستريت وما اعطاني رياكشن، بعرف انه الدي اي بي اف اجى وعمل انهبيشن لهالانزيم، سو دايركتلي بقدر اعرف انه عندي serine residue involved in the interaction between the enzyme and the substrate. بتقولوا لي اوكي بس فيني اعرف قد ايه رقمها، اي سيرين اي 195 سيرين 250 هون بهالميثود لا، بس بعرف انه في سيرين. I can identify the amino acids. I have to do for sure additional method لروح اقدر اعرف اي هي كانت involved in this interaction. So the IPF, it's a group specific reagent. You have a lot of serine residues in the chymotrypsin, but the DIPF, we will see later on, they identified the serine involved, the serine 195 located in the active site of the enzyme where the DIPF was able to inactivate the serine. Later on, by partial hydrolysis, أكيد بس صار عندكم هالinteraction, you have to do a partial hydrolysis of the EI complex that will give you a series of peptide fragments which can be separated, analyzed, and the amino acid sequence of these fragments will be, you will do the sequence and then you can identify the amino acid sequence where you have the interaction between the inhibitor and the enzyme, which is the chymotrypsin here. And they found the sequence glycine aspartic acid, serine, glycine, glycine, proline, and the serine residue in question was the serine 195. So the IPF can also inhibit other enzymes غير يعني كل شيء هلا ما قلنا قبل شوي انه كل enzymes اللي هن serine proteases اللي عندهم السيرين in the catalytic triad of the active site منهم التريبسين الالastase السبتيليزم الاسيتيلكولينستريز thrombin وكلهم serine proteases and the DIPF is able to inhibit all these enzymes. So all the enzymes belonging to the serine protease category اللي عندهم serine involved in the catalytic triad are able to be inhibited by this group specific reagent اللي هو DIPF. منهم اهم شيء نسمعهم كثير الاستيز تريبسين السبتيليزم ثرومبين اسيتيلكولينستريز chymotrypsin etc. Do you have any question? So بحيالله exercise او بحيالله analysis عم تعملوه او شفتوا انه بالجيفن حطوا لكم الدي اي بي اف وصار في عندكم انتم automatically an inactive complex او ما صار في reaction, automatically one of the conclusion you can do is that you have for sure a serine present in the active site of the enzyme.
دكتور يعني هاي الأمينو أسيد بتكون involved in the binding؟
مين عم بيحكي؟
سارة.
سارة هلا later on رح نفهم شوي هلا ما عم بقول لك وين موجودة. later on عرفتي انه في serine involved. اوكي؟ you have to do your reaction وتشوفي KM وتشوفي catalytic والVmax. okay, and then these kinetic parameters will help you to know هل amino acid when can involved based on the modification of the kinetic parameters اللي هن KM and K catalytic.
كده.
اوكي لانه انت كيف حتعرفي اذا بالبايندينج ولا بالكاتاليزس؟ based on the modification of the Km and K catalytic values. صح؟
صح. اوكي.
so هول اللي رح يحددوا لك وين موجود. انا هلا عم بعطيكي ميثودز لاقول لك كيف تعرفي نوع شو هو الامينو اسيدز اللي ممكن يكون انفولد. اوكي؟
اوكي دكتور، ثانك يو.
So, enzyme modification experiments also have revealed histidine, cysteine, serine, methionine, tyrosine, aspartate, glutamate, lysine, tryptophan residues by using group-specific reagents, some of the amino acids located in the active site of the enzyme. And here we will see other examples than the DIPF. For example, في عندنا iodoacetate. Iodoacetate is an important group-specific reagent that is known to react with a histidine. So, alkylation of histidine residue by iodoacetate was shown to inactivate the ribonuclease, for example. So here, the enzyme with iodoacetate, which is here the ribonuclease, أعطانا carboxy methyl enzyme as complex, اللي هو كمان inactive. By using this method, they were able to identify that histidine 119 and to a less degree another histidine اللي هو histidine 12 may be alkylated by the iodoacetate, but one of them, but never both at the same time. يعني يا اما يعني عملوا experiment مرة يلاقوا الهستidine 119 واوقات يلاقوا الهستidine 12 بس ولا مرة اثنيناتهم at the same time كانوا alkylated. So شو يعني؟ يعني these two histidine وهذا it's also a lot of experiments can obtain same results. This means that these two histidine residue are close to each other in the active site. هلا iodoacetate عم بيعمل interaction مع واحد منهم مش مع اثنين. اوكي؟ اي الاكسسبل له اكثر 19 ولا 12 بيعمل معه interaction. So هول اثنين هن present in the active site واثنيناتهم لانه عم بيطلع عنا على طول واحد منهم هو اللي عم بيعمل interaction مع iodoacetate. So حكما اثنيناتهم they are very close to each other in the active site. مش ضروري يكونوا in the primary structure بس in the active site عنا هول اثنين. فطبيعي بس عم بيكون السبستريت عم بيجي يعمل interaction يمكن عم بيجي يعمل interaction مع هيدا ريزيديو او هيدا ريزيديو. يعني بكفي واحد منهم يعمل interaction مع substrate for the reaction to start or to take place. So they noticed that these two histidine are located in the active site of the ribonuclease. They are close to each other and the substrate to react with one of these for sure. هلا iodoacetamide another reagent than iodoacetate. iodoacetate اللي هو ICH2COH. iodoacetamide is a similar alkylating reagent used also but has no effect on the enzyme. Okay, so they noticed that the negative charge of iodoacetate may be able to form a electrostatic interaction with the protonated form of one of the histidine residues prior to the carboxymethylation of the other residue. So iodoacetamide was not able to, which is also an alkylating reagent, but was not able to inactivate the enzyme in this case. however, iodoacetamide can inactivate enzymes with a fuel group. يعني إذا عندنا enzyme with cysteine amino acid as involved in the active site, iodoacetamide is able to inactivate the cysteine to form a inactive enzyme. So iodoacetamide عنده activity مع الcysteine. عندنا iodoacetate with histidine. مثل ما شفنا قبل كمان ال DIPF with a hydroxy of serine. So هول الاشياء اللي لازم تحفظوها وتعرفوها. Other example هو ال PCMB parachlorometry benzoate. ال PCMB also is able to interact with dual group of a cysteine to form a inactive complex. This such reagent attacks the dual group in a non-specific fashion, يعني هو منه specific. Okay? And that's why usually when we use PCMB in a reaction طلع عنا inactive complex, you have to do ممكن تكون cysteine, okay, but you cannot confirm, okay, and you have to do further reaction to validate this point. And usually في عنا ال NEM, ال N-ethyl methylamide, which is a confirmation نعملها لح confirm your results with the cysteine, and using an M they were able to identify the cysteine 25 of papain involved in the catalytic pain, the active site of the enzyme, to form inactive complex. So usually بس نعمل PCMB ويطلع inactive complex to confirm this as cysteine, they can go and do the interaction with NEM that will also get a inactive complex. Okay. Other reagents also used as group-specific reagents هن DTT-AB, dicarboxy diisothiocyanate azobenzene. It's a bifunctional reagent. يعني bifunctional إذا بتطلعوا بالstructure هي عندها مثل similarity in the middle. This reagent binds with NH2 of 2 lysine residues, but for sure these 2 lysines should be also close enough. So بس يكون في عندنا doubt إنه 2 lysine present in the active site close to each other, you can use the TTAB as a molecule with bifunctional group to interact with the 2 lysine involved in the active site if these 2 lysine are close enough to each other to inactivate the enzyme. So in this table you have a lot of molecules انتم المسؤولين عنهم هن بس اللي حكينا عنهم هلا قبل شوي as examples, but in this table you can see a lot of reagents. They are specific with a specific amino acid side chain. So we'll not go in all examples of group reagents, but just those with the examples we gave you previously in this slides. But just to know there is a lot of other molecules to interact with specific amino acid, specific side chain of a amino acid. هلا في شيء كثير مهم لازم تعرفوه انه sometimes you have a order of reactivity for these amino acids. يعني ممكن يكون في عنا amino acids يعمل interaction with more than one reagent. One molecule can interact with more than or could be can interact خلينا ما نسمي شيء ثاني with more than an amino acid بس حيكون عنده order of reactivity for these amino acids. For example, iodoacetamide or iodoacetate هن عندهم by order of reactivity with the cysteine first, then tyrosine, then histidine, then lysine. يعني هن ممكن يعملوا interaction مع هول الأربع amino acid. بس إذا في cysteine وتيروزين الorder of reactivity بياخذهم تيعملوا attack مع الcysteine أكثر من التيروزين التايروزين اكثر من الهيستidine مع الهيستidine اكثر من اللايزين. او اذا عندنا الفليورو 24 دايناميك اوف رياكتيفيتي مع السيستين اكثر من اللايزين اكثر من التايروزين اكثر من الهيستidine. So for sure sometime you have a molecule that can react with different amino acids, but what you should know that they have an order of reactivity. يعني بحال انه for example هيدا reagent اشتغل مع histidine شو يعني انه بهالاكتف سايت ما كان عندي سيستين وما عندي تايروزين، لانه لو كانوا موجودين كان اجباري يعمل رياكشن معهم قبل الهيستidine. So this order of reactivity will help you to exclude يعني شو ممكن يكون ما كان موجود بهالاكتف سايت. اوكي، رح نوقف هون اليوم، بنكفي الخميس هذا second part of this chapter ونبلش exercise بال double substrate. Do you have any question before we move to the exercises?
دكتور
تفضلي.
ميليا، بس الستراكشرز مسؤولين نعرف أشكالهم.
Yes منهم كتار دومازين.
اوكي ثانك يو.
إذا بنراجعهم شوي رح أقول لك عندك DAB بس ومنه صعب لأنه هو المولكيول مقسومة قسمين، منه كتير صعب لتحفظيها. NEM يس، عندك PCMB يس، وحتشوفيهم هلا بالاكسرسايز. انا حقلك اكتب الرياكشن حتعرف انه مطلوبين. عندك اليودو اسيتاميد، اليودو اسيتيت، وفي عندنا قبل كان الدي اي بي اف. اوكي، يعني منهم كتار، مش هلا اللي مارقين بعدين مش حيكونوا كتار، هول اللي مرقوا معنا اليوم، لانه ممكن اقول لك اكتبي الرياكشن. اوكي، شو بصير انتراكشن بين بين الانزيم وهول الرياجنتس. Other question? طيب يعني مين حل الاكسرسايز؟ ميريا؟
يس دكتور.
يا تفضلي. بدك تعملي نمبر 2؟
يس بقرا السؤال. يلا.
تفضلي.
We measured the initial velocity of an enzyme-catalyzed reaction using different concentrations of substrates. The concentration of one substrate X is first held constant and present at different concentration of other substrate Y, and vice versa. Results expressed in micromole per minute are shown in the table below. Determine all kinetic parameters you could obtain from both primary graphs without drawing any secondary graphs. هون هو عطينا 3 values أصلاً. رسمت أول primary لما بتكون ال X constant.
Draw all 3 هنا 10.3 وبدك ترسموا 3.
Yes. طلع السكيل هينا 10 مايكرومول minus 1 دوت minute، وعلى الاكس اكسس 0.1 مايكرومولر minus 1.
هون طالب منك
الاكس
to determine all kinetic parameters you should obtain from both primary graphs without drawing any secondary. بس طالب تدرس مول primary.
يس الانترسكشن طلع على الواي اكسس.
لحظة خلينا
يعني
نشوف أول شيء اليونت موزيل 1 over v. شو حطيتي يونت لها؟
مايكرومولر minus 1 دوت minute.
Okay. E is expressed in micromolar per minute. So here micromolar minus 1 dot minute will x رسم الواي الأول micromolar. So 1 over y micromolar.
يس
For this
والاكس constant.
concentration v max طلعت constant.
true.
Okay. So second
بتاني جراف
primary.
ال1 على v طلعت مايكرومولر minus 1 دوت minute كمان، بس ال1 على x هون ملي مولر minus 1 منحية هون بال
ملي مولر minus 1 لأنه الإكس هو ملي مولر.
Yes.
Okay.
From primary graphs we can obtain kx minus 1 over kx طلعت معنا minus 1.4 millimolar minus 1 هيها. Yes. فالkx equal 0.71 millimolar. من السكند بلوت من أول واحدة قصدي if x equal هون اعتبرت ال 5 infinity دكتور، ما بعرف إذا true هالشي.
مين قال لك؟
لأنه إنه لقيت كتير في فرق، يعني وحدة 5 وحدة 0.67 وحدة 0.33.
أو إذا مين قال لك أعلى فاليو هي انفينيتي؟ ما فيكم
ما بعرف هيك.
تعملوا انفينيتي إلا إذا هن بالتيبل قايلين لكم 5 حاطين لك حدها تنس تو انفينيتي. هي ساعتها فيك تعتبري انفينيتي، بس إذا ما عاطينك هيك no you are not allowed to use the highest concentration as a value that is standing to infinity.
اوكي فكرت نحن لازم نستنتج.
اوكي سو من البرايمري بلوت جبتي من وحده الكي اكس، من اول وحده شو فيك تجيبي من اول بلوت؟
الفي ماكس.
في ماكس. So the primary plots will give you from the first one 1 over v as function 1 over y will give you the value of v max. 1 over v max constant قديش؟
1 over v max طلعت معنا هيها 12. فبتطلع الفي ماكس equal 0.083 micromolar per minute. سألت yes. Second question, deduce the order of binding for both substrates. What is the kinetic parameter that you were not able to determine in this exercise? It's ordered sequential mechanism. It's ordered
sequential mechanism. Yes.
At rapid equilibrium, since where x bind first, where x bind first, because plot where x is variable and y constant is intersection is outside the axis to the left of y axis.
صفر.
لا تحت سويت
بس هيدي الجملة ما بتقوليها.
ما بنقولها كمان. اوكي.
لا. You have to discuss based on the value of the max and Km with the different concentration of the constant molecule. ما فيك تقولي since the interception is outside the axis تاخدي صفر أو on the axis.
Okay. For x increase, plot where x is constant, 1 على V max apparent equal 1 على V max minus 1 over K.
On the y variable x constant, okay?
Yes.
Okay.
1 over V max apparent equal 1 على V max minus 1 over Km decrease. فال 1 على Km increase يعني affinity increase.
Okay.
For y increase, هاي البلوت where y constant. 1 على V max apparent less than 1 على V max, so V max increase. Minus 1 over Km decrease, 1 over Km increase, affinity increase. اللي ما قدرنا نطلعهم parameters K prime X and K prime Y وال KY كمان. Parameters can't be fixed.
Do you have K prime in a ordered mechanism? دموزيل ميليا.
Yes. نو لا ما عنا.
اكتبي إذا
أنا
بدك تكتبي
غلطت.
ما أنت فيرست، ما هيك؟
إيه أنا غلطت سوري. الكي واي بس اللي ما قدرنا نطلع.
إيه. أوكي، هو ما عندك لا كي برام إكس ولا كي برام واي. بس
صح.
أنت هون ناقص كان الكي واي، جبت الكي إكس وجبت في ماكس، ناقصك كي واي اللي كان فيك تجيبيها من سكندري بلوت.
صح.
أوكي؟
Yes.
Yeah, thank you. Number 3.
Welcome doctor.
مين بده يحل number 3؟ Number 3. بعدنا ناطرين؟ ما حدا؟ مين اللي حليتيه أنت number 3؟
إيه لا دكتور بلشت فيه بس ما طمنت.
شو يعني بلشت فيه؟
هلا الصبح يعني بلشت برسمة أول جراف بس.
والباقي ما حدا راسم شيء؟ ما حدا؟ نو؟
دكتور أنا عملت الجرافس بس ما صورته.
ما فيك تصوري. إذا لا بنوقف هون والخميس بنعمل كورس وبنكفي اكسرسايز. بس ما بيمشي الحال هيك اكسرسايز بالنهار. بعد عنا 3/4 ساعة مينيموم ممكن لازم نعمل 2 اكسرسايز today.
دكتور أنا حلي 7 إذا
7 ما في شيء بعد 7. 7 كورس اوكي. 7 و 8 كورس ما من شيء. عملي 7 يعني. مين اللي قال لي؟
آميل. بس دكتور بليز تعطيني أكسس ترجع.
يلا هي 7 هو بيور كورس يعني منه
Yes, doctor.
يلا تفضلي.
Establish the secondary graph for mechanism represented by rate equation below. Explain your answer in detail. V equal V max over 1 plus KB over B plus KA KB over A times B equal V max times B over B plus KB plus KA times KB over A. مثل ما شفنا ال V max equal constant, then we must use KB apparent in the secondary graph. ال KB apparent versus B or 1 over KB apparent versus 1 over B. ال KB apparent equal KB plus.
لا إيليا انت
Yes.
شو عم تقول لك؟ ايه apparent versus شو؟
versus versus A
1
versus A هون غلطة بالكتابة.
1 over A مش A.
Yes.
لانه
Yes.
طلعوا كلكم على الجيفن. هلا كان فيهم يعطوكم بس انتبهوا كان فيهم يعطوكم to establish the secondary graph for the mechanism represented by the rate equation below. Explain your answer in detail. يعطوكم بس اول وحده ما يكفوا equal شيء. يعطوكم V equal V max divided by 1 plus KB over B plus KA KB over AB. هلا هون انتم كفولكم ثاني ضربوا ببي لانه هون كان بي فاريبل واي كونستنت، ضربوا ببي فوق وتحت صار عندنا بي اوفر بي بلس كي بي بلس كي اي كي بي اوفر اي. هلا هون صارت واضحه انه في ماكس كونستنت ايزوليت بي في شيء اسمه فوق ايزوليتد. صار عندي انا انه بي هي كي بي بلس كي اوفر اي كي بي، يعني كي بي as common 1 plus KA over A أو KB apparent KB أو 1 over A KA KB plus KB. وبيطلع عندنا هون السكندري بلوت اللي لازم ترسميها function of 1 over A. اوكي بس اذا بتوسعي لهم شوي الصوره تاعيتك لانه كثير صغيره مش مبينه، اذا بتقدري تعمل شويه زوم. اوكي سو كي ام apparent لبي هي كي بي بلس K over AKB، يعني أنت عندك هون عم ترسمي KB apparent as function of 1 over
as function
A.
of 1 over A. صح.
أوكي.
السلوب هي KA KB، ال Y intercept هو KB وال X intercept هو minus 1 over KA. لكن بس هون هي KB apparent versus 1 over A.
صح.
اوكي. شو ثاني؟
عاملة ال8 وراها؟
نو دكتور.
هذا البين بوين يعني كمان لا مش بين بوين. اوكي. طيب any other exercise؟ عشان عاملين بس 1 و 2 7 هالسات. لازم نعمل at least الخميس مع الكورس 3 و 4 و 5. سكس كمان هو هايين 6 فهم. طيب يلا بنوقف هون اليوم، فمش حالين شيء بس بليز بدنا حدا يشتغل. انا ما بحب الاكسرسايز، بدنا حدا يشتغل بليز. اوكي يلا هاف ا نايس داي، وهلا بشوفكم برجع بالابيديميو بعد شوي.